Bacteria secrete several exoenzymes that hydrolyze large molecules. Hydrolytic enzymes break down large substrates with the addition of water. The resulting small molecules can then be transported into the cell. Amylase hydrolyzes the polysaccharide starch into glucose. Amylase is commonly used to distinguish species of bacillus. We will test 3 bacteria for the production of amylase using a starch agar plate. The medium contains starch and also peptone to support growth of bacteria that don't use starch. Iodine is added to the medium to test for the presence of starch. You can see in the spot plate that iodine is brownish. The iodine turns blue when mixed with starch. Divide a starch agar plate into thirds by labeling the bottom of the plate. Mark the underside of the plate to divide it into thirds. Then write your name, the date, and the test. Aseptically, streak a single line of bacterium 1 onto 1/3 of the plate. Flame your loop until it is red hot and let it cool. Use the loop to obtain the first inoculum. With your loop, streak a single straight line from the middle of the plate into the first sector. Inoculate the second section with the second bacteria. Flame your loop and let it cool. Use your loop to obtain the second bacteria. Turn your plate clockwise for a quarter turn and streak a single line through the second third of the plate. Inoculate the third section with the third bacteria. Flame your loop and let it cool. Use your loop to obtain the third bacteria. Turn your plate clockwise for a quarter turn and streak a single line through the last section of the plate. Incubate the plate, inverted at 35 to 37 degrees Celsius for 24 to 48 hours. Record the presence of bacterial growth. One of our bacteria didn't grow, so we can't determine this bacterium's ability to hydrolyze starch. Using the plate with bacterial growth, flood the plate with grams iodine. Make sure the iodine covers the entire plate. You can tilt the plate slightly to help distribute the iodine. Iodine only binds to non-hydrolyzed starch, turning it blue to brown in color. If amylase is released, the starch will be hydrolyzed, so a clear zone adjacent to the bacterial growth will appear. If there is no clearing, then the bacteria did not produce the amylase enzyme. You can see that the starch was hydrolyzed outside of the cells, indicating amylase is an exoenzyme. This bacterium hydrolyzes starch. Starch is visible up to the amylase negative bacterium. Dispose of the plate properly.